bacteria are a diverse group of gram-positive non-spore-forming microbes widely used in food fermentation, probiotics, and biopreservation due to their ability to metabolize sugars into lactic acid. Key genera include Lactobacillus, Lactococcus, Streptococcus, and Pediococcus. These bacteria are categorized based on their metabolic pathways, habitats, and applications. For example, in the developing infant gut, microbial colonization begins immediately after birth and follows a structured succession. The environment is initially oxygen-rich, favoring facultative anaerobes such as E. coli, Streptococcus, Thermophilus, and Lactobacillus species like Rhamnosus and Fermentum. As the gut matures and becomes more anaerobic, Bifidobacterium species dominate, especially in breastfed infants due to their unique ability to digest human milk, oligosaccharides. In adult guts, microbial diversity significantly expands. The gut becomes fully anaerobic, and nutrient input diversifies to include complex carbohydrates, proteins, and fats, which allow more niches and interactions. Bifidobacterium, while still beneficial, plays a less dominant role and often engages in cross-feeding relationships. For example, its production of acetate supports the growth of plantarum and butyrate-producing species. Culturing these strains requires Lorodox, oxygen-free media, often supplemented with reducing agents like L-cysteine. In contrast, strains like Lactobacillus plantarum are generalists, capable of utilizing diverse carbon and nitrogen sources and tolerating low pH, making them ideal for robust industrial fermentation. While the workflow can be universal for designing a typical fermentation experiment, the strain-specific characteristics uniquely determine their culture media, environmental condition, and especially the experimental objectives of why adopting them, which in turn determine the culture formats and monitoring strategies in the experiment. Microbial experiments differ in goals, but whether you're producing a metabolite or validating an analytical method, the objective determines what to monitor, how to measure it, when to sample, and where to culture. 5 common experimental goals can be either product, strain, process, function, or method-oriented. When the goal is to maximize product yield or quality, such as improving yogurt flavor, increasing probiotic viability, or enhancing metabolite output, the monitoring strategy acts like a quality control system, directly tracking parameters tied to end product performance. In yogurt fermentation, using Streptococcus thermophilus pH that defines which influences coagulation and mouthfeel and aroma compound levels are key. Sampling intensifies near the fermentation endpoint when flavor compounds accumulate. For EPS producing rhamnoses, monitoring can focus on viscosity and polysaccharide yield, while in bacteriocin producing lactis, antimicrobial activity and concentration are usually quantified. Monitoring the acidification rate and final pH of Pediococcus is essential to ensure the texture and safety of fermented sausages, while fermentomy is often tracked to produce flavor-enhancing metabolites. When the goal is to study inherent microbial traits under specified conditions, such as growth kinetics, stress resistance, or metabolic pathway preferences, the experiment becomes a physiological investigation. This often requires multiple replicates for reliable data. Comparing Bifidobacterium strains for acid bioresistance uses microplate assays to assess cell viability. And considering their oxygen sensitivity, close monitoring of OD or key metabolites helps pinpoint their growth limits. When the goal is a consistent, high-efficiency fermentation, whether for biomass, enzymes, or bioactives, real-time control of the culture environment and its follow-up optimization are critical. In high-density cultures of GF Patagti coli, dissolved oxygen should be carefully managed alongside real-time fluorescence to guide induction and nutrient feeding. If the objective is to test the function of a bioactive compound, monitoring goes beyond quantity. It should confirm biological efficacy. To assess the broad-spectrum antimicrobial activity of Reuterin produced by Reuteri, samples taken during production can be analyzed not only for concentration, but also via agrodiffusion or microdilution assays to determine inhibition zones or minimum inhibitory concentrations against pathogens like E. coli. Lastly, in method-driven experiments, the microorganism serves as a testing platform to evaluate the performance of analytical tools. The primary focus is on the accuracy, precision, sensitivity, and robustness of the method itself. In summary, depending on the experimental objectives, strain characteristics dictate the focus that shifts among our adjustments that center around factors such as strain growth rate, metabolite levels, environmental conditions, and the measurements taken. Besides, the strain characteristics guide contamination control to ensure result consistency, accuracy, and the safety throughout experiments. Lactase is highly susceptible to bacteriophage infection, necessitating the addition of calcium citrate to the medium to chelate calcium ions and suppress phage absorption, along with regular plaque assays to screen for phage presence. Notably, B. subtilis itself can be a source of contamination as its spores exhibit high resistance to standard autoclaving at 121 degrees, thus requiring extended sterilization times of up to 45 minutes to ensure complete inactivation. E. coli, by contrast, although it does not form spores, its rapid proliferation in nutrient-rich media allows it to quickly outcompete target microbes in sterile systems. Contamination often manifests as unexpected turbidity, color change, or pH drift. Therefore, aseptic technique is fundamental and essential in executing microbial experiments and protecting both personnel and the environment. In other words, all manipulations with the strains must be conducted in a laminar hood with surfaces and gloves disinfected using 70% isopropanol before and after handling. Pipette tips and inoculating tools should be sterilized via flame or autoclaving. Proper personal protective equipment, including lab coats, gloves, masks, closed-toe shoes, trousers, and safety goggles, is mandatory to prevent exposure to airborne spores or potentially irritating metabolites. Finally, the liquid waste requires treatment with 10% bleach for at least 1 hour before disposal, while solid waste should be sealed in certified biohazard bags to be autoclaved. By the way, never discard non-biohazard materials, such as flammable solutions like isopropanol, into biohazard bins, as this poses a serious fire hazard.